Hello, it's a pleasure to be speaking today at the Little Steps virtual conference. My name's Mark Diamond. I'm the Managing Director of Antisense Therapeutics. Antisense Therapeutics is based in Melbourne, Australia, and we're focused on developing up our pipeline of antisense drugs. We have two drugs in our pipeline. We call them Atl1102 and Atl1103. And of course, today I'll be focusing on our drug Atl1102 that we have been testing in Duchenne's muscular dystrophy, where we've been able to generate some very, very encouraging preliminary data in non-ambulant boys with Duchenne's. So the slide here overviews Duchenne's muscular dystrophy. For this audience, I suspect these details are well known. What I'd just like to focus on, though, is that the point that our drug, Atl1102, is being designed to treat the inflammation in the muscles that's associated with the loss of dystrophin. Today, the only way really of being able to address that inflammation in these boys is through the use of high-dose corticosteroid. And we know that when used chronically, the corticosteroids can have some significant side effects, including weight gain, reduced bone density, and other retardation side effects. So we believe there's an important need for a superior treatment to the use of corticosteroids for the inflammation in the boys with Duchenne's. And we think Atl1102 is profiling with exciting therapeutic potential to address that inflammation and do it in a superior and safer way to the corticosteroids. Atl1102 is an antisense drug that's designed to block the production of a protein that's expressed on the surface of white blood cells or lymphocytes, the proteins known as CD49D. And CD49D is responsible for allowing the white blood cells to bind to blood vessel walls and migrate from the blood into sites of inflammation. So our drug looks to block its production, thereby stopping the aberrant conduction or movement of these lymphocytes from the blood into inflammatory sites, such as the muscle tissue in patients with Duchenne's. What we know also from some important clinical work undertaken by a team in France and Brazil, Dr. Pinto Mariz was the lead author on this publication in skeletal muscle in late 2015, where they observed that Duchenne's boys that had more severe and rapid progression of their disease had more of this CD49D on their lymphocytes. So they were known as being high expressing of the target CD49D. And what's important also to note is that the corticosteroids don't appear to have any effect on CD49D expression on the cells or the number of cells with that are expressing CD49D. So we know that our antisense drug is working differently too, a unique mechanism and over the mechanism of action of the corticosteroids. And that's why initially we've been testing our drug in combination with the corticosteroids. We're the only company in the world that has a drug targeting CD49D in development for Duchenne's. And what we also know is that boys, the non-ambulant boys, as they're more advanced with the disease, have more of the CD49D, the target of our drug, on their lymphocytes. So we ran our first clinical trial in non-ambulant boys with Duchenne's. We ran the study here at the Royal Children's Hospital in Melbourne, where we tested the drug for 24 weeks of administration at once weekly in subcutaneous injection of 25 milligrams. The study was primarily a safety and tolerability trial, but we were also keen to assess the drug's effects on disease progression in these boys. And of course, in the non-ambulant boys, the primary way of checking on the drug's effects on disease progression is through the assessment of their upper limb functional capacity and strength. And we use the standard assessments of PUL2 and MySet to get a sense of what the effect the drug was having on upper limb capacity. Firstly, very importantly, we're able to show the drug was well tolerated and safe in the study. We had no patients withdraw from the trial. We had no serious adverse events reported. So very happy with the safety profile of the drug at the dose tested. We, though, were also delighted to report on the activity benefits that were observed in this study. We saw that Atl1102 showed an improvement in muscle strength when you're looking at the mean changes over the 24 weeks of therapy versus the known and losses of boys who are non-ambulant assessed over the same time period. We were also able to show in the study that 7 of the 9 boys saw either a meaningful improvement or stabilization in their PUL2 score versus, again, the expected losses in that population over the same time frame, even when the boys are on their corticosteroids. Underpinning this data was also some very exciting observations around the drug's positive effects on fat fraction in the muscles. We're able to show a stabilization in fat fraction and a preservation of functional muscle mass. So observing the musculocellular positive changes backing the very pleasing and highly encouraging physical benefits that we observed in this study. You'll see here we've included on the slide a quote from our key opinion leader, Duchenne's expert, Professor Thomas Voigt in the UK, and his views on the very positive nature of these preliminary findings. In October of this year, we had data presented at the World Muscle Society Congress. It was a poster presentation where our PUL2 data from our trial was compared to an external matched natural history control. So these were a group of boys that had been monitored by an expert in Europe, in Italy, Professor Eugenio Mercuri, who was observing their disease progression and monitoring their PUL2 changes. So we're able to match our study population with a matched population of boys in his database. You can see here that we saw a mean improvement in PUL2 versus the substantial losses in this cohort of boys, even though these boys were on their maintenance doses of corticosteroid. Again, we've included Professor Mercuri's views on the data in this quote provided here. Important to note that PUL2 is what's regarded as a key efficacy endpoint for seeing approval of drugs in non-ambulant boys with DMD. So again, very, very, very excited by this preliminary data, particularly now when we're able to compare it to a matched control. As I said, next phase of development for our drug is a planned phase 2B clinical study that we're looking to run in Europe. Quite advanced with our plans for this phase 2B clinical trial. All going well, we're expecting to see submission for a trial approval first half of next year and approval to conduct the study middle of next year. In parallel, we're well advanced in plans for defining the clinical path and next steps for the drug in the US. We have now orphan drug designation in the US. We also have rare pediatric disease designation in the US, and just recently had reported positive opinion around orphan drug designation in Europe. So we're making very solid progress. It's a very important juncture for the company, and I'm being delighted today to be able to present on our plans and our data to date, and look forward to updating the DMD community as we advance towards our phase 2B clinical trial. Thank you.